Hi guys, welcome to our last day of Bio 148. So today I'm going to be out in my front yard because I want to show you guys a few things and I think that it'll be a lot of fun to play with genetics. So it is our last day. You've gotten through it, day 18 for content. So I know that that is so fast and we've gotten through everything so fast. Next week you guys will be working really hard on your projects and studying for your final cumulative exam, and I'll give more information about that later, okay? But for today, let's focus on some fun genetics, okay? So I'm out in the front yard because it has a few cool things out here. So we just have some coneflowers starting to bloom, and my neighbor has some that are yellow, and so let's just play with the genetics of them. We're gonna make it all up, but let's just play with the genetics of them and make sure this is on. Okay, and see what would happen if we cross them. Oh look guys, there's a little bee on it. Okay, maybe we'll play with some bees too if we find more than a bee, one bee. Okay, so let's start a new note. I've had to record this a few times because I've had tech issues, so let's see if it'll work this time. Okay, all right, so let's see, we are on day 18. Let's do pink because that coneflower out there is pink. Okay, so day 18. And we're working through genetics, okay, and inheritance. And so we're working through the inheritance of alleles, figuring out what's dominant, determining dominance, and using Punnett squares to predict ratios of offspring when we're given different genotypes, okay? So we have here my pink coneflowers. So pink coneflowers. Nope. There we go. And then I also have a neighbor that has yellow ones. Okay, so let's say that I want to know if mine or my neighbors are dominant, okay? And one thing to note is that all of the flowers that pop up in my yard, when I have like new ones, they're all pink, okay? And my neighbors are all yellow. And so when this happens, we call this pure breeding, okay? So, oops. So both mine and my neighbor have pure breeding lines. And this is when offspring always look identical to their parents, right? And in order for that to happen, it must be homozygous, right, for a certain trait, okay? So here I have pink or yellow coneflowers, and let's think about this for a second. If I have pure breeding lines, right, I don't know what the genotype is going to be. I just know that they're going to be the same, right? So I'm going to say that my gene of interest here, okay, is going to be the color gene. And I have a capital C, so one of these two is capital C's, and then the other one is lowercase c. I like to use that little slash through to keep track of what's the dominant and what's recessive, okay? And right now I don't know which one's dominant or which one's recessive. So let's say that I take some pollen from one and cross it with the other one and make an offspring, okay? So let's say that when I do this, all of the offspring are pink, okay? So all of the offspring are pink. And this individual, when I've crossed two individuals together, okay, that are pure breeding, I know that I am going to have heterozygous, okay? So these individuals are called F1 hybrids. And F1 hybrids are always heterozygotes. So that means that this individual is big C, little c, okay? So this helps me determine dominance, okay? So the second thing that I will do is use the heterozygote individual, to determine dominance. okay? So here my heterozygote is pink, right? So that means that the pink allele is the one that's dominant, okay? So big C equals pink, right? So big C in my pure breeding line, that had to be homozygous, versus the yellow is going to be recessive. Okay, so that means my neighbor's yellow one is a recessive trait. I don't actually know if this is true, okay? I just am playing, okay? So just keep that in mind. I'm just making this up. Okay, so the things that I'm not making up, though, are that F1 hybrids are always going to be heterozygotes, and that will help you determine dominance, okay? Because just one copy of this C allele is going to make it so that your whole individual is pink, okay? Okay, so then we can do lots of other things after this. Maybe we want to cross two F1s or do some other cross. So cross two individuals. And to do this, we have to first determine the gametes. Okay, so remember that our gametes can only get one homologous chromosome. It's not going to get both of these, okay? So if I'm going to cross two F1s together, big C little c by big C little c, okay, this individual can give big C or little c. Oops. Okay, this individual also can give either big C or little c. And now I have my Punnett square, okay? So I can drop down the, like, egg and sperm, and like male and female isn't quite how plants do reproductive isolation, but just go with me here, okay? Since that's what we've talked about before, okay? So from the egg, we'll get the big C, okay? Or the little c. From the sperm going over, we can get the big C, or we can get the little c, okay? So these are my different options. And so what you'll see in these squares are the zygotes, okay? So my zygotes are in the squares. So to do this, right, I determined my gametes first, and then I found the zygotes, okay, and by filling in the table, okay? So notice here that for my gametes, I only have one copy of the gene, right, because they're haploid. The gametes are haploid, right? Gamete. Versus in here, the zygotes have two copies. They're diploid. Okay? Okay, so now I want to maybe figure out what my genotype ratios are, okay? So maybe the fourth thing I want is genotypes. So here, the ones that I care about, right? Maybe I want to look at homozygous dominant, heterozygous, and homozygous recessive, okay? So I want to figure out my genotypic ratios. And that's just saying how common each one is, okay? So here I have one that's big C, 2, 1, 2, that are big C, little c. So note that in this case, the big C came from dad and the little c came from mom. Down here, the big C came from mom and the little c came from dad, okay? And then I've got one that is little c. So note that the homozygous ones are the most rare because you had to get both big C from mom and dad in this case, or little c from both mom and dad in this case, okay? And then maybe I also care about phenotypic ratios. So when you're looking at, I'm going to actually switch this to genotypic ratios. So when you're looking at your zygotes, you can figure out the genotypes as well by using your knowledge of which one is dominant, okay? So when we go up here, we know that big C is dominant, so anything with at least one big C is going to be pink, right? Big C is pink, okay? So big C is pink. Here we have one big C, so we're going to have pink. One big C, so we're going to be pink. Here we have two little c's, so we're going to be yellow, okay? So both of these, right, are going to be pink. So my phenotypic ratio for pink is 3 to 1 yellow. And that's what I'm going to, that's what I'm going to expect in the offspring, right? So if I have 4 offspring, I'm going to expect that 3 of them will be pink and 1 of them will be yellow, right? If I have 12 offspring, then I'm going to expect 9 of them to be pink and 3 of them to be, did I do that math right? And 3 of them to be yellow, okay? So you can figure out what your expected amounts are in your offspring, okay? And now I might not tell you to cross 2 F1 individuals, or maybe I won't have you start with pure breeding lines. Maybe what I'll do instead for something like this is I'll say that you have, maybe I've already done the genetics on it, and I could tell you that you have a pink heterozygous individual, and you're going to mate it to a yellow homozygous individual. Okay, now the yellow homozygous doesn't tell me what the genotype is, right? But the pink, right, does. So when it's heterozygous, I know I have big C and little c. So this information right here, right, pink heterozygous tells me that big C equals pink, right? And then that tells me also that the yellow homozygous is little c, right? So from this pink individual, I could get either the big C or the little c. So I can make my table like this, big C or little c. From the yellow one, I'm only going to have this little c as an option, right? This parent will only give 1, right, in the gamete, but it only also has 1 option, okay? Okay, so that means I've got big C, little c from the mom, little c always from the dad. So in this case, my ratio is actually going to be 50-50, right? So 2 little c's equals yellow, and over here, 1 big C equals pink, okay? So I'm going to have a 1-to-1 ratio in this case instead. So 1 pink to 1 yellow. I also only have 2 genotypes here, right? So one big C, little c to 1 little c. So if this is what I'm crossing and I have 10 individuals that are made, I expect 5 of them to be pink and 5 of them to be yellow, okay? Okay, so we can do this with single genes, but we can also do this with when we're looking at 2 different genes. So let me go back outside. Okay, so we can also do this when we have 2 different genes. So here, let's look at my blueberry bushes. Okay, so here are my blueberry bushes, and this blueberry bush here has a ton of blueberries on it right now, okay? It's a couple years old, and it is doing well and has a lot of blueberries, and they will ripen in mid-July, okay? Over here on the side, this side, I have another blueberry bush, and you can see where it had blueberries on it already, okay? Because it is already done for the year. So there's 1 more left that's still ripening, but for the most part, they're all gone, okay? So it had fewer blueberries, but they all ripened in June, okay? So let's play with that, okay? So let's play with this idea of my blueberry bushes. So one of them, let's call it that first bush that still has the blueberries on it, that are still kind of greenish in color, okay? So bush number 1, okay, had lots of berries, and they're going to ripen actually in July, okay, versus bush number 2 had fewer berries, but it ripened in June, okay? And I don't know here which one is dominant, which one's recessive, okay? But I do know I've got 2 different genes. One is controlling how many berries, and the other one's controlling the timing, okay? So the first thing that I'm going to do again is to figure out my different traits, okay, of interest. So my different traits. So one is going to be the berry gene, either a few or a lot. I don't know which one's dominant and which one's recessive right now, okay? Nope. don't want to do that. Okay, so this is berry number. And then the other one is timing. So when it ripens, okay? So big T and little t. And again, I don't know which one's dominant and which one's recessive yet. So I'm going to cross the two of them, okay? So if they're pure breeding lines, I can cross them and get the heterozygotes. So my F1 hybrids, okay? Let's say that I have lots of berries in June, okay? So my F1 hybrids, remember, are going to be big B, little t, because they are heterozygous for all of the traits of interest. Lots of berries. And let's say that that happens in June, okay? F1, wait. Okay, so lots of berries in June. So this lots of berries is big B, little b. The June is big T, little t. So that tells me that lots of berries is dominant and a few berries is recessive. And then for timing, it tells me that June is dominant and July is recessive, okay? So now I can fill in what these parents were. So lots of berries was big B. And remember, if I'm in pure breeding lines, they're going to be homozygous. The timing for July was recessive, okay? Bush number 2 has big T, let's go back. Little b, and big T, okay? Because it is recessive for the berry trait and is dominant for the timing trait. Now, in some cases, it's not that you have to have one that's dominant and the other one's recessive. Maybe they could have both been dominant for bush 1 and both be recessive for bush 2, okay? This is just what my F1 hybrids told me, okay? Okay, so now that I figured out dominance, I could do a cross, right? So I could do a cross of interest of some kind. So maybe I can cross 2 of these F1 hybrids together, okay? And this is called a dihybrid cross, okay? Because it's 2 genes and I'm crossing hybrids, okay? Up here when we did the 2 hybrids, F1 hybrids for the color, this was called a monohybrid cross where I had 1 gene. Okay. Okay, so that's just a side note for when you get to genetics. Okay, so a dihybrid cross. So here I'm going to cross big B, big T, little t, individual by big T, big B, big T, little t, individual, right? So I need to first find the gametes, okay? So to do that, I need to think about how I'm going to FOIL, right? So I have big B, big T from 1. I have big B, little t. Okay, I've got little b, big T. And I have little b, little t. Okay, and we're going to do the same thing for the other one. So I can have big B or little b, big T or little t, right? So the first options are big B, big T. Okay. Then the outsides, right? So big B, little t. Then the insides, little b, big T. And the last, little b, little t. Okay. Okay, and now I can fill all of this in. Oops. Okay. And okay. So I'm gonna just drag all these big B, big T's down. The next column, big B, little t. And I like to give space when I have a recessive trait for the dominant trait to fit in, okay? Then I've got little b, big T. And then finally, little b, little t. Okay. And then I'm going to drag over these ones. So big B, big T. The next row is big B, little t. The third row is little b, big T. And then the last row is little b, little t. Okay. Okay, so now I want to know about the phenotypic ratios. I don't really want you guys to worry too much about the genotypic ratios here. We can talk about that again when you get to genetics, okay? So I just want you guys to worry about the phenotypic ratios. So remember our phenotypes, right, are lots of berries or few berries in either June or July, okay? So I like to do the one that's dominance first, or both dominant first. So that's lots of berries in June, okay? So lots of berries in June, right? So that is going to be big B and then something, and then big T something, okay? I could also have lots of berries in July. So that's big B something, little t, right? Because I might have to be recessive for the timing one. I could have few berries in June, right? So it's a little b. I need both homozygous recessive to get the few berries, and then big T whatever to get the berries in June. And then I could also have few, oh, what is that? I don't know what's going on. Okay, few berries in July. So it would be little b, little t. Okay. So here are all my different options of things that I could have, right? Whether I have a lot of berries or few berries, whether they're in June or in July, and I know my genotypes for them, okay? So now I need to try to find these genotypes up above so that I know what my phenotypic ratios are going to be, okay? So if I'm looking at this first for my dominant traits, okay, they only have to have been given by one of the parents, right? So they're going to be the most common traits that I can find, okay? So if I'm looking for a big B, big T, I'm going to find that really frequently in this Punnett score, okay, in this table. Okay, so let's plug it in. So let's do this as a different color. So let's do lots of berries in June in green. Okay. In green. Okay, and let's see. So I need a big B. It doesn't matter if I got it from mom or dad, and a big T. Okay. A big B and a big T. A big B and a big T. Big B, big T. Big B, big T. Right, so that first row, I got big B, big T from dad, right? This first column, I'm going to get big B and big T from mom, right? So I know that all the way down this row, I'm going to have the big B, big T combo, okay? And then I'm going to have somewhere I get like, I could get big B from mom and big T from dad. So right here I have that combo. Or I could get big B from dad and big T from mom. So here's another one, okay? So let's count how many of these we have. One, oops. Okay. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, okay, that are going to be big B, big T. Okay, now I'm looking for big B in little t. So lots of berries in July, okay? So let's look for lots of berries. Actually, let's use a color that isn't in our background. Okay. So lots of berries in July. Oops. Go away. Okay. So if we've got lots of berries in July, let's find big B, whatever, and little t. So I need big B, little t. Big B, little t. Big B, little t. Okay, so I've got 3 of them that are like that. 3 that have that combination. Next, I'm going to look for few berries in June. So let's look for few berries in June. So I need little b, and then big T, okay? So little b from mom, little b from dad, and then a big T. I don't care who gave it to me. Big B from, little b from mom, little b from dad, and then a big T. Little b from mom, little b from dad, and then a big T, okay? So I've got 3 of those again. So I've got 3 of those. And then finally, let's look for the few berries in July, okay? So the only one that has few berries in July, okay, is going to be this one right here, okay? That's the only one that's going to be few berries in July. So I only have one of those, okay, where I have few berries in July. Okay, so my total number of squares here, I've got 4 by 4, right? Because I've got 4 different gamete options here at the top, right? 1, 2, 3, 4, and 4 different gamete options also from dad along the side, right? So I have 16 different zygote options, right? So this should total 16. So 9 plus 3 is 12, plus 3 is 15, plus 1 is 16. So that's like my check that I did everything correct, okay? I also want you to realize here that the number that is the most is going to be when we have both traits being dominant, okay? When we have that 3, that intermediate number, because one of the traits is dominant and the other one is recessive, right? Same thing here, only which trait is dominant, which. Here's recessive and here's dominant. And then this last one is going to be the most rare because I had to get both recessive alleles from both parents, right? Up here, right, when we were looking at like this purple one, so here I could get big T from mom, right? I could get big T from mom and dad, or I could just get big T only from dad, right? So there's 3 different ways that I could get there, right? Versus in this one, I had to get the recessive one from both parents, okay? Okay, so that's why it's most rare, is because I had to get recessive from both parents. Okay, again, I could ask you this question this way, where I'm giving you kind of a phenotype and you have to figure out the hybrid and go through finding dominance that way, and maybe I have you do a dihybrid cross, or maybe I tell you something else. Maybe I tell you that I have 2 blueberry bushes, and, oops, let's say that bush 1, I know has lots of berries in July, and I know that the lots of berries one is heterozygous, and I know that the July part is homozygous. Maybe I've done some genetics on them, okay? But I don't actually know which one's dominant and which one's recessive, right? Kate. You can figure out this out though, right? So you know that the het is going to be big B, little b, right? So if that one is big B, little b, then I know that big B is lots, right? And little b is few, okay? This timing thing, I don't know yet, okay? This thing about July, I don't know, okay? So maybe I tell you, okay, that bush number 2 has lots of berries and is homozygous. So you can already fill that in. You know that's big B, big B. And I tell you that it is heterozygous, or the timing one, in June. Okay? So that tells you which one's recessive and which one's dominant for your timing. So this tells you that June is dominant, right? And so July must be recessive. So that's little t, little t. Okay, so now you could cross these. Maybe you want a certain phenotype that you don't see in either of your parents. Maybe you want a few berries in June or a few berries in July, okay? And so if you cross these together, right, bush number 1, let's do little t there. Okay, bush number 1 can be big B or little b, and the little t, right? So you can get, because you can only get little t from this one, right? You can't get the big T. So you can get big B, little t, or you can get little b, little t, right? For bush number 2, you can only get big B, right? And you can get either big T or little t, right? So you can get big B, big T, or big B, little t. And then you can do your Punnett square to figure out phenotypic ratios, right? So big B, little t, little b, little t, little b, little t, big B, big T, going that way, big B, big T, big B, little t, big B, little t. Okay, so now you can figure out, okay, well, if I have these same combinations of things, right? Let's grab all of these, but not the ratios, okay? Copy that, pull it down. I can try to figure out if I can get all 4 of these combinations, or maybe I can't, okay? So let's look for something that is big B and little t, right? So I need, oops, there we go, big B and big T, big B and big T is right there. Big B and big T is right there. So I have 2 of them that have this dominant phenotype. Then let's look next for the big B and the little t. So it looks like I've got 1, 2 of those as well. So 2 of those, oops. Okay, and then I don't have any actually of these other 2 at all, okay? So my phenotypic ratios are going to be different if I've crossed something like this instead. So you always have to figure out the gametes first, right? These ones are those ones. Find out the gametes first and then figure out the zygotes that belong on the inside, okay? Okay, so I will do a separate video on the learning objectives here in just a few minutes.